really a great honor to address this audience. I would like to tell you about the work we are doing to understand how cells make proteins, more specifically how cells make proteins fold into their correct shape, what I call the origami of the cell here. I was told that there are several non-biologists in the audience, so I'm trying to explain what we are doing in a way that some of you at least can understand the basic principle, and I apologize to the experts for simplifying a few things. Now, this project was started as a purely basic science project. We just were curious to understand how an important process in biology works. But as you will see, it turned out that there are important implications of what we found with what we just heard from Professor Sheckman regarding the neurodegenerative diseases such as Parkinson's disease. Now, proteins come in many shapes and sizes. This wallpaper shows you a nice collection of different proteins. They are responsible for almost all the functions in our cells. They do the work. They are required for catalyzing chemical reactions. They make our muscles move. They make our brain cells work. And of course, they transport the oxygen in our blood. So they do almost everything. And in order to do that, the newly synthesized protein chain consisting of amino acid building blocks has to first fold into an intricate and well-defined three-dimensional pattern. This is what I call the origami. The main finding that we made was that this process requires pre-existent protein machines in the cells that we call molecular chaperones. And here is what a guest professor from Japan told me would come close to what Japanese would understand by molecular chaperone. I hope it is correct. I think it is supposed to be the molecular version of an assistant in an important ceremony such as a wedding. And that's quite similar to what these chaperones do at the molecular level. They help the newly made protein chains to fold. Now, this protein folding reaction happens after the polypeptide chain has been made on the ribosome. The ribosome is the machinery that puts the amino acids together. You can perhaps imagine it a little bit like a typewriter that puts letters together, but these letters are amino acids. And each protein chain in our cells has a specific sequence of these amino acids. And this sequence is encoded in the DNA. It is transcribed into the messenger RNA, which the ribosome uses as a template to put the amino acids together. So it comes out as a linear chain, but then has to fold up into this intricate three-dimensional pattern. And this step is required for the protein to have its biological function. Now, as you can see here, proteins are extremely small, approximately in the range of 5 millionth part of a millimeter. So this origami that we are talking about is very complex and much smaller than the paper folding origami. So what we found is that this process requires these molecular chaperones to be effective and to occur at a relevant, biologically relevant time scale. So here's the comparison with your paper folding, Japanese paper folding. Now, in the presence of the molecular chaperone, the amino acid chain, amino acids just like pearls on a string, will fold into the correct functional state. Protein is active. But what happens in the absence of these machineries, or when they are dysfunctional, as it happens in some of the diseases, then proteins misfold and they clump together. And these clumps can be toxic to cells. We'll get back to that in a moment. Now, this would be similar to an origami folding attempt gone wrong. Now, how can we imagine such a protein aggregate? Many of you, many of us, in fact, are looking at one every morning when we have our breakfast. And this is a fried egg where the white part of the egg is essentially a macroscopic protein aggregate. Due to the heating process, the liquid protein, which is the folded protein, the egg white, has unfolded, and the unfolded protein chains stuck together. So having something like this in your cells, of course, would not be good. Now, how does the chaperone machinery avoid this from happening? I'm showing you here an example of the major chaperone system that we have been studying for many years, called HFSP, which is a cylindrical protein complex. So chaperones are proteins themselves. And it captures the unfolded, not yet properly folded protein after it has been delivered by another chaperone called HFSP. So an important thing we learned is that different types of molecular chaperones cooperate in defined pathways, and these pathways have been highly conserved in evolution, from bacteria to yeast cells to human cells. Now, something really amazing is happening in the sense that this chaperone cooperates with another factor called HFSP10, which is basically a lid that encapsulates the partially folded protein inside the central cavity formed by the HFSP. And now we have only a single protein chain inside this cavity, and is allowed to fold for a couple of seconds, which is timed by the conversion of ATP molecules to ADP. ATP is the energy currency of the cell. And once the lid opens, the folded protein emerges and can now fulfill its biological function. It's a little bit simplified, but that's the basic idea. A single protein cannot aggregate with other proteins while it folds. Now, I'm illustrating this here in a movie, hopefully it will start, where you can now see how the mRNA is decoded on the ribosome. The polypeptide chains, the amino acid chains are emerging from the ribosome and will now be protected by this first set of chaperones, the HFSP molecules, and will then be transferred into the central cavity of the HFSP cylinder, the barrel-shaped molecule that allows the protein to fold in its interior. It should come into view in a moment. These are the domains of the ring that are sticky, and they bind to sticky surfaces on the not yet folded protein. These are hydrophobic amino acids. And it is encapsulated and now allowed to fold on its own inside this nano cage, which, when it opens, releases the folded protein. Now, as I alluded to before, we then learned in the course of these investigations from our work, and of course from the work of many other groups worldwide, that these chaperone systems have an important medical significance. These chaperones are required to keep the protein universe of our cells, millions of protein molecules, in an intact shape and functionally active. They constantly survey these proteins, and if they find that something is wrong with them, they first try to refold them, to repair them, and if that fails, they transfer them to degradation machineries. There are machines in our cells that can shred these proteins back into amino acids. Now, what we have seen is that during the aging process, unfortunately, the capacity of these chaperone machineries undergoes a decline. And this is now believed to be facilitating the manifestation of some of the diseases Professor Scheckman just talked about, including the Parkinson's disease. Now, this is a major socio-economic and medical problem. Here is a graph that illustrates the projected numbers of Alzheimer's disease patients. At present, we stand at about 7 million in the US alone, with an expected doubling by 2040. And there are also approximately 1 million Parkinson's disease patients at this point. In total for Alzheimer's and Parkinson, I was told about 4 million in Japan. So this is a huge problem. In all of these diseases, what happens is that these protein clumps form, the protein aggregates, either outside or within the cells. And when we look at these types of aggregates, we find that they generally have the shape of so-called amyloid fibrils. They have an elongated fibrillar shape in all of these different diseases. So these deposits form in the brain cells, and they are apparently damaging the brain cells. The chaperone systems prevent this from happening for the most parts of our life, but at some point they no longer have sufficient capacity to do so. That is one factor that probably plays a role. Now, cells can make more chaperones when needed, for example, under various stress conditions, such as a fever state, for example. When temperature goes up, some proteins will unfold and possibly clump together. But as I said, the functional capacity of these systems undergoes decline during aging. Now, as a result of this, we then have an imbalance created between the production of misfolded proteins and the available chaperone capacity. And our goal, of course, now would be to rebalance the system, to maintain these chaperone systems and other machineries of protein quality control in a more useful state, possibly by treating people with a drug that upregulates these chaperone machineries. This has been possible in cell culture experiments, but not yet in patients. But I'm hopeful that by better understanding these systems, we will be able eventually to manipulate them in such a way as to result in new therapeutic strategies. Thank you so much.